Hello everyone, I'm Jianping Zhang, currently work at CSIRO, Australia. It is a great honor for having this opportunity to share some of our most recent works on weight stem rust resistance with you. As said in the title, today I will talk about the cloning of SR9 locus, the largest stem rust resistance allelic series in wheat. Wheat is an important crop for it provides about 20% calories for human being daily. And wheat rust disease has been the major threat for wheat production since Roman times. Historically, farmers used to rely on the superpower to worship the god of the wheat rust. Host resistance was used to control rust started as early as the 20th century and following by the renowned Green Revolution, which was initiated by Dr. Norman Borlaug. One of the major components of Green Revolution is actually the breeding for resistance against stem rust. The well study and utilization of SR9 were part of these efforts as well. With 8 alleles predicted at a single locus, SR9 becomes the largest stem rust resistance allelic series in wheat. The only other comparable one is the PM3 for powdery mildew in wheat. I used to wonder why I'm often hearing about SR2 but never SR1. So what is SR1? It is actually also one of the SR9 allele, SR9D. In fact, the first SR9 allele, SR9A, was reported as early as in 1956. And in 1960s, we got 2 more. 3 more from 1970s. The most recent 2 alleles were reported in 2014 and 2021, respectively. I'd really like to give a shout out for all these early wheat scientists. Definitely a real tour de force, spans over 60 years. Based on these excellent previous works, and taking the advantage of all the recent developed bioinformatic approaches and the whole genome sequencing progress in wheat and its relative species, we successfully performed the isolation of all the previous reported SR9 alleles. It is a synergistic strategy combining genetic mapping, allele mining, target sequencing enrichment, and exome capture. Here, I would like to use SR9B as an example to briefly describe how we capturing the target gene candidate. For SR9B, we screened 7 loss of resistant mutant lines, and by comparing all the NLR type genes with the wild type, we identified a candidate gene with all the loss of resistant mutant lines that containing either missense or nonsense mutations that resulted in the losses of gene function. As this is a strong indication of this gene is responsible for the given resistance. So by using similar approach, we cloned all the other alleles. So what do we learn from our cloning work on SR9 locus? Firstly, we found these SR9 alleles are orthologs of the SR21. As we can see from the phylogenetic tree, the SR21 is the closest SR gene to SR9. Secondly, there were 2 SR9E haplotypes. SR9E1 is from Weinstein, and the H2 is from Volna-Emma. These 2 haplotypes differed at 8 amino acids. Last, we found the SR9F allele, which was reported from Chinese Spring, is actually not SR9 allele. And this was supported by the Chinese Spring sequencing data, as well as the resequencing of our own and original Chinese spring genetic stock. This figure shows all the identified and confirmed SR9 alleles from our study. The lower bar is the indication of the percentage of sequence conservation. As we can see, all these S9 alleles shared a highly conserved N-terminal, and this high conservation even extended to the NBRC domain and part of the LRR domain. And the most polymorphic region was identified towards the C-terminal of the LRR. To further validate the gene identity, we also performed the transgenic complementation experiment. As we can see from the tree, the SR9 alleles are clustered into two subgroups. So we took one allele from each subgroup as representative. As expected, the T1 transgenic plant displayed similar level of resistance in comparing with their wild type. So for SR9H, the wild type we're using is Gable 56. And in the case of 9EH2, we actually used the Field plus 9EH2 instead of the original source of 9EH2, the Chronos, because Chronos have another SR gene, SR13. These works were carried out by Namala and Matt from USDA, Shisheng and Hongna from Peking University. Here comes the most interesting discovery from our study. Taking the advantage of the recent available AI-based protein prediction tool AlphaFold, we are able to obtain the high confidence full-length protein structure prediction for all the SR9 and the SR21 proteins. Surprisingly, we found all the SR9 allele together with SR21, all having the unusual long LRR domain that contains 42 LRR unit. Here from 1 go around up to 42. And compared with the majority of the rest SR protein only having 17 LRRs, we also confirmed that the 3 alleles of SR9H, SR9B, and SR9G differed only at 2 residues. If we take a closer look, the 9H and 9B, and the 9B with 9G, actually, pairwise, they have only 1 amino acid difference. For instance, this glycine to arginine change actually enabled the SR9H resistance against the UG99. It is noticeable that both 9H and 9G differed with SR9B for 1 single positive charged residues, which is methionine to lysine and glycine to arginine. You will be very fascinating to explore how these positive charged residues were involved in altering the RAVR recognition or the resistance specificity. Not only the unusual LRR, these SR9, SR21 allele encoded proteins also have a unique N-terminal structure. We found, unlike the rest of the cloned NLR type SRs, all the SR9, SR21 proteins are lacking of a predictable coil-to-coil or cis domain. And both the N-terminal sequence alignment and the structure prediction agreed with this feature. we identified an extra 51 amino acid motif only from the SR9 and the SR21 proteins. And this extra motif appears to form also an extra helix when compared with the typical 4 helix bundle CC domain previously reported for SR33, SR35, and Arabidopsis R proteins R1. So to test whether this unique N-terminal structure would carry out the similar function as the cis domain for SR33 and SR50, we expressed the equivalent fragment of SR50 from SR21 and SR9H. And the result confirmed, although having these differences, the equivalent SR21 and SR9H fragment both can trigger cell death in benzimiana, same as SR33 and SR50. So before I list the take-home message for my talk today, this background with all the countries labeled for all the contributing institutions is to show that this project is not only a perfect example as a relay of scientific legacy of the WIT researchers, but also global synergistic collaborations and efforts. To summarize, we isolate most of the SR9 alleles with multiple strategies. We confirmed 5 of them by either loss of function mutants or transgenic complementation. And we found SR9 alleles are orthologs of SR21. We identified the SR9H9B and the SR9G9B proteins are distinguished by only 1 amino acid. And although all the alleles of SR9 encoded highly related NR-type proteins, however, they confer a unique resistance spectrum. And last, comparing with other cloned SR proteins, the SR9 and SR21 proteins may contain the different N-terminal structure and a Euro long LR region. I'd like to acknowledge all the colleagues who have contributed to this study, especially Colin from Agriculture Canada, Bob and Peng from the University of Sydney, Plant Breeding Institute, Cobbetty, George from UC Davis, and Matt from USDA and the University of Minnesota, as well as Evans from CSIRO, for their initiating and conceiving this project. All the funding bodies supported this research are well acknowledged as well. So given the time limit, if you would like to have more information about study, you are more than welcome to join me online. And thank you so much for your time.